Welcome to the Deep Dive. Today we're taking a high-velocity shortcut through a pretty complex stack of sources, all focused on one really fascinating molecule, 5-amino-1-MQ.
Right.
And for you, the listener, who's probably constantly sifting through new research in metabolism, well, this compound has been described as a genuine metabolic switch.
It's definitely generating some serious conversation, and for good reason, I think. Our mission here isn't just to define 5-amino-1-MQ.
No.
We really need to understand its unique mechanism. Why does it seem to sidestep the usual rules? You know, the rules about appetite control, hormones, CNS stimulation, all that stuff that comes with many weight-related compounds.
Exactly. So the mission today, understand what makes this mechanism different. The sources really stress at this point, this compound doesn't change your behavior. It doesn't suppress appetite. It doesn't make you feel jittery or anything like that. Instead, it seems to fundamentally reprogram how your cells actually use the energy they already have.
And that reprogramming, it happens deep inside the cellular machinery. It's not like when people hear metabolic compound, they often expect maybe a stimulant or something targeting a hormone.
Right.
5-amino-1-MQ is neither of those. It's more like a cellular optimizer. It works on one specific target to essentially, well, take the foot off the brake in your cell's metabolism, just lets it run more efficiently.
Okay, so optimizing the engine itself, not changing the driver, not adding like high-octane fuel, but just making sure the fuel delivery system isn't leaky. That's a good way to put it.
Pretty much.
So let's start by unpacking exactly what that brake is.
Okay. To really get 5-amino-1-MQ, you first have to understand the specific enzyme it goes after. Nicotinamide N-methyltransferase. Thankfully, we can just call it NNMT.
NNMT.
Gotcha.
Got it. So this is the metabolic bottleneck you mentioned.
That's the one. It's the bottleneck we need to talk about.
Okay, so why is NNMT considered a bottleneck? Let's dive into what it normally does. We know it acts on nicotinamide. Yeah. That's a key form of vitamin B3, right? Yeah.
Exactly. Vitamin B3. Most people know B3 is important, but NNMT's role is quite specific.
So what's it doing to it?
Well, NNMT's natural job is to act as a kind of regulator, especially in, say, fat and liver cells, though it's found pretty much everywhere. Its function is to permanently modify nicotinamide.
Modify how?
It performs a process called methylation. Basically, it sticks a small chemical tag, a methyl group, onto the nicotinamide molecule. And once that tag is on, well, that nicotinamide becomes effectively useless for the cell's main purpose, making energy.
Ah, okay. So it's not destroying the B3 molecule itself, but it's just shunting it down a dead-end street, taking this really valuable resource, a building block for NAD, the cell's energy currency, and just saying, nope, this one's basically trash.
Precisely. It's a diverting resources. Nicotinamide is supposed to be recycled and built into NAD. But when NNMT gets its hands on it, it transforms it into something else, a metabolite called 1-methylnicotinamide, or 1-MNA.
And then what happens to that?
It gets kicked out of the cell, excreted. The sources use a great analogy comparing it to a fuel tank leaking.
Right. You've got all the fuel coming in, but a chunk of the most critical part of that fuel is just being systematically wasted, leaked away.
And the research seems to indicate that this NNMT activity, it actually ramps up, right? It gets more active in states like obesity or diabetes.
That observation is absolutely key, yes. It suggests this sort of wasteful pathway gets accelerated when there's metabolic stress or, just too much caloric input. So NNMT isn't just some baseline thing. It seems to be part of the actual dysregulation that happens when cells get overwhelmed. It becomes a way the cell maybe paradoxically stops itself from maximizing its energy efficiency.
Okay, so if NNMT is the enzyme creating this leaky, inefficient system, how does 5-amino-1-MQ actually stop it? How precise is this countermeasure? Is it like a shotgun or a sniper rifle?
Oh, definitely a sniper rifle. The precision is really the elegant part of the chemistry here. 5-amino-1-MQ is designed as a highly selective competitive inhibitor.
Meaning it kind of mimics the shape of nicotinamide just enough? Just enough to physically sit in NNMT's active site. That's the exact spot where NNMT would normally grab the nicotinamide to tag it.
Ah, okay. So it doesn't destroy the enzyme or anything drastic. It just parks in its parking spot, basically.
Yeah.
Renders it inactive while it's sitting there.
Exactly that. It's not some systemic poison or a metabolic sledgehammer. It's like that very precise wrench that locks one specific gear, NNMT, while letting all the others run smoothly.
Okay.
And the selectivity is absolutely crucial because, well, cellular metabolism is incredibly complex, right? If an inhibitor just messed with dozens of different reactions, you'd get chaos. Side effects galore.
And the sources really emphasize that selectivity, don't they?
Mhm.
They say it doesn't disturb other enzymes in that whole big NAD/GAP pathway. It doesn't mess with the enzymes that make NAD, only the one specific enzyme that wastes its precursor.
That is the power of targeted inhibition. By putting a very specific block on that one waste mechanism, the cell immediately starts conserving the nicotinamide that was previously just getting flushed out.
Right.
And that conserved nicotinamide, it's now available to feed straight back into the NAD production pathway. So you raise the cellular energy budget, basically immediately and efficiently. That shift from waste back to resource, that's the whole game with 5-amino-1-MQ.
Okay, so we've effectively unclogged that metabolic bottleneck by blocking NNMT.
Yeah.
Now, let's talk about the payoff. What happens immediately? We see a rise, or at least preservation, of two really critical molecules. You call them the fuel currency and the repair budget.
That's right. The first one, as we touched on, is nicotinamide adenine dinucleotide, NAD. This is like the coenzyme of coenzymes. It's absolutely fundamental. NAD is essential for converting the nutrients you eat, glucose, fats, whatever, into ATP, which is the main energy molecule the cell actually uses. But it's more than just fuel. We need to be a bit more specific for our audience.
Right. It's more of the shuttle service, isn't it? NADIX is this crucial carrier for hydrogens and electrons, like currency exchange inside the cell.
Good analogy.
To break down fuel, the cell uses NADE to pick up these hydrogen atoms and electrons, turning NAD into its other form, NADH. Then that shuttle, NADH, takes those electrons over to the mitochondria, the power plants, to generate, well, huge amounts of ATP.
Exactly. And when NNMT is highly active, it's basically starving the system of the raw material, the nicotinamide, needed to make that NAD.
So it slows down the whole shuttle service.
Right. Therefore, when 5-amino-1-MQ blocks NNMT, the cell can suddenly make more NAD, or at least preserve what it has. And that dramatically improves the efficiency of that whole energy production line. More NAD available means faster, more complete energy production. Simple as that.
Okay, that covers the energy side. Now let's look at the second molecule, which honestly seems like maybe the most unique part of this whole thing. SAM S-adenosylmethionine. Why does saving SAM matter so much, and how is it connected to NNMT?
SAM. Okay, SAM is integral to literally thousands of biochemical reactions. It's the cell's primary universal methyl donor.
Methyl donor, meaning it gives away those little chemical tags we talked about.
Precisely. Those methyl groups are like little flags needed for all sorts of critical jobs, cellular repair, turning genes on or off, detoxification, just general metabolism. SAM is kind of the cell's repair and maintenance budget.
Okay, and here's the kicker, right? The connection. When NNMT tags nicotinamide, it doesn't just waste the nicotinamide. It has to get that methyl tag from somewhere.
And it gets it from SAM.
So NNMT is a double drain.
Exactly. It's a dual resource drain. It depletes the nicotinamide pool, lowering the potential to make NAD, and it consumes SAM, draining the cell's capacity for all those essential methylation reactions. So NNMT activity is really like a double whammy against optimal cellular health.
Wow. Okay, the consequence of blocking NNMT then is, well, it's pretty profound. You simultaneously boost the energy potential by saving NAD precursors, and you strengthen the repair and maintenance systems by saving SAM. It's not just a partial fix. It's like a systematic upgrade across two absolutely critical pathways. The cell's natural energy production can thrive, like the sources say, and its ability to just maintain itself, its integrity, is also boosted.
And look, we don't have to just rely on theory here. The lab evidence is actually quite compelling. Researchers took adipocytes fat cells and treated them with 5-amino-1-MQ.
What did they find?
Well, they observed a predictable and pretty substantial increase in the NADU content inside those cells, which directly confirms that waste prevention mechanism is working.
Okay, so NADU went up, but what about the metabolic result? What did the fat cells do differently?
That was the real kicker, as you said. In those same fat cells, the actual production of new fat lipogenesis just dropped sharply.
Right.
Yeah. The cells, simply by having higher internal efficiency, by having more NAD available, they fundamentally changed how they handled energy storage.
Okay, let's connect that to the bigger picture. If a cell is flush with NAD, what does that encourage it to do?
Well, generally, it incentivizes the cell to shift its energy handling towards oxidation, meaning burning fat and sugar for immediate fuel rather than just storing it.
Right. Use it or lose it, essentially.
Sort of. So this preservation of the vitamin B3 precursor and the subsequent boost in NAD helps cells produce energy more efficiently. It translates to this overall metabolic tune-up, makes the cell run cleaner, prioritize spending energy over just hoarding it.
Okay, so now we move from the Petri dish, where we saw fat production drop in isolated cells, to the studies in whole organisms. And here's where it gets, I think, really interesting, because we run into what the sources are calling the behavioral disconnect.
Ah, yes. This is the finding that really sets 5-amino-1-MQ apart from, well, almost every other compound currently targeting weight loss.
Yeah, it is it.
The animal studies, particularly those using obese mice, fed a high-fat diet. They showed that giving these mice daily doses of 5-amino-1-MQ caused significant fat loss and reduction in body weight,
Okay.
even though the mice kept eating the exact same amount of food.
Wait, let's just pause on that for a second. That's kind of huge, isn't it? In a world that's so dominated by the simple calories in, calories out model,
It really is.
these animals did not reduce their calorie intake at all, yet they significantly lost body mass, and specifically fat mass. That seems like core evidence that this compound is genuinely reprogramming metabolism from the inside out, rather than just making them eat less.
Absolutely. It strongly suggests that 5-amino-1-MQ is primarily acting on caloric partitioning, basically dictating where the calories go once they're actually in the body.
So instead of just shunting extra energy straight into fat storage.
Right. Instead of that, the optimized cells seem to be diverting that energy towards, say, heat production, maybe increased movement, or just a higher baseline metabolic rate. In essence, burning it off before it has a chance to settle into storage.
The energy output changes dramatically, even when the energy input stays exactly the same. What specific data points did they find that confirm this shift, this move from storage to expenditure?
The specifics are pretty illustrative. Beyond just the drop in total body weight, the analysis showed the treated mice lost significant fat mass. That's key. And when the researchers actually looked at the adipose tissue, the fat itself, they found that the existing fat cells had physically shrunk in size.
Wow.
So this wasn't just, water weight or maybe muscle loss. This was targeted fat reduction.
Okay, shrinking fat cells. What about other markers? Did anything else change systemically?
Yeah, they saw broader improvements too. The treated animals showed decreased cholesterol levels, which points towards a generally healthier systemic lipid profile.
Good.
And importantly, reinforcing that cellular mechanism we talked about, the compound actively suppressed lipogenesis, the creation of new fat in those lab-grown cells we discussed earlier.
So it's doing both, shrinking existing fat stores and preventing new ones from forming.
That seems to be the powerful dual action, yes.
Okay, let's circle back to the mechanism. If the animal is eating the same amount of high-fat food, the only way it can lose mass is to burn more energy, right?
Correct.
So how does having higher NADO and SAM translate directly to an increased burn rate?
Well, it ramps up the processes involved in fat burning, what scientists call fatty acid oxidation. With more NADO available, the cell essentially has more capacity for the enzymatic machinery needed to break down fat molecules.
So you're giving the mitochondria, the power plants, the resources they need to handle more traffic.
That's a great way to think about it. More NADO means the power plants can run harder and faster, processing more fuel. So 5-amino-1-MQ seems to effectively flip on the body's internal burn fat switch. But crucially, it does this by ensuring the cell's energy budget is healthy enough to prioritize using fat as fuel. It dictates energy expenditure through biochemistry, providing a solution that, fascinatingly, doesn't rely on the user having to, force themselves to skip that slice of cheesecake.
That is a profound finding. It really challenges our sort of singular focus on purely behavioral interventions for weight management.
And of course, this optimization of NAD isn't just happening in fat cells. Muscles are some of the most energy-hungry tissues in the body, right? They rely heavily on super efficient mitochondrial function.
Absolutely.
So it makes sense that the benefits would extend beyond just fat metabolism, potentially improving muscle performance and recovery too.
Okay, let's shift focus there. The performance data, especially in those models looking at aging, the studies with older mice and muscle strength seem particularly striking.
They really are quite remarkable. Researchers looked at aging mice, which naturally start to experience sarcopenia, that's age-related muscle wasting and weakness.
Yeah.
The older mice treated with 5-amino-1-MQ showed approximately, get this, 40% greater grip strength compared to the untreated older mice.
40%.
Yeah.
That's That's huge. That's a massive functional improvement just from a metabolic tweak.
It is. It suggests they were, in a way, metabolically younger, or at least functionally more robust. Their muscles were just working better.
And the sources also highlight this synergy with physical activity. 40% is impressive just from the molecule itself. What happened when they combined the treatment with exercise?
Right. So when they gave 5-MeO-1-MQ and had the mice follow a physical activity regimen, the improvements were amplified. They saw about 60% greater grip strength compared to the sedentary control group.
60%?
Yeah. And this is crucial because it indicates 5-MeO-1-MQ isn't just acting as like a replacement for exercise. It seems to be an enhancer of the adaptations from exercise.
Okay, explain that.
It suggests that the optimized muscle cell, with its better fuel system, thanks to 5-MeO-1-MQ, can actually reap far greater rewards from the effort being put in. The exercise stimulus leads to a bigger improvement because the underlying machinery is running better.
It's like having a perfectly tuned engine during a race. The car's still being driven, the effort's still there, but it just handles the stress and performs so much more efficiently.
Exactly. Now let's talk about recovery, which is so critical, especially for aging and just maintaining mobility.
Okay, the regeneration study, what did that show?
This one focused specifically on injured muscles, but in very old mice, a group that really struggles with repair processes.
Right, healing slows down a lot.
It does. But in this model, the muscles in the treated mice regenerated dramatically more effectively after injury. And the quantifiable results really underscore this. Analysis showed that the recovered muscle fibers actually became nearly twice as large as the fibers in the control group's repaired muscles.
Twice as large, meaning the muscle was rebuilding itself much more robustly, more completely.
Yes. And functionally, their strength after recovery increased by about 70% compared to controls in that injury model.
70% recovery in strength.
Yeah. And this ties directly back to those core molecules, NAD and SAM.
How so?
Well, higher NAD levels support the massive energy demand needed for the repair process itself. And SAM provides all those crucial methyl groups required for gene expression changes and protein synthesis, basically, all the molecular instructions and building processes involved in rebuilding the tissue. It's providing both the energy and the key molecular signals for repair.
Okay, this all naturally leads us towards the longevity angle, doesn't it? If NNMT levels tend to rise with both obesity and aging, like the sources suggest, what are the implications of blocking it for potentially a healthier lifespan?
That's the big question, isn't it? The implication is that maybe the aging process itself is partly characterized by this systemic increase in metabolic wastefulness driven by NNMT. So blocking NNMT could potentially support healthier blood sugar regulation because more efficient cells handle glucose better and maybe help maintain a state of more youthful cellular metabolism for longer.
And by optimizing NNAO, you might activate those longevity pathways we hear about, like sirtuins.
Exactly. Sirtuins are absolutely dependent on NADA to function. So boosting NADA availability by reducing waste could indirectly activate these protective pathways. It suggests that maybe just preventing this internal resource drain could be a powerful tool for maintaining vitality as we age.
It's a compelling idea.
Yeah.
But we absolutely must be responsible here, right, with our interpretations.
Absolutely critical, yes. While the underlying mechanism points towards these really interesting anti-aging and maybe insulin-regulating benefits, we have to stress the crucial caveat again.
Which is?
Direct human evidence for these longevity effects, or even for the blood sugar effects in humans, and certainly for long-term safety in humans, it's still completely pending.
Right.
These are fascinating observations from robust preclinical models, cell cultures, mice, but they are absolutely not clinical guarantees for people. Not yet.
Okay, so we've established the specific biochemical power of 5-MeO-1-MQ, selective NNMT inhibition, conserving NAD and SAM, leading to better metabolic efficiency. Now, for you, the listener, who's trying to make sense of all the metabolic compounds out there, this is vital. We need to contextualize this. When you're looking at your sources, how does 5-MeO-1-MQ actually fit into the bigger picture? How is it different?
This is really the learner's shortcut, isn't it? If you understand that how the mechanism, you can sort through the information much more effectively. We need a clear sort of mechanism by mechanism comparison to show why 5-MeO-1-MQ is unique and not just, another flavor of existing therapies.
Okay, let's do it. Let's start with the current giants in weight management, the GLP-1 analogs like semaglutide. How does a behavioral modulator like that stack up against a biochemical optimizer like this?
Okay, V's, GLP-1 analogs. Right. GLP-1 agonists, they work primarily by hitting receptors in the brain and the gut that control satiety.
Fullness.
They make you feel full. Exactly. They make people feel full faster, eat less, so they significantly reduce food intake. They are powerful behavioral interventions. They change the input side of the equation.
Well,
5-amino-1-MQ is totally different. As we've seen, it works inside the cells, changing the output side, the efficiency of the engine. There's zero evidence it affects hunger or satiety signals, so the mechanisms are completely distinct pathways. One is neurohormonal, affecting behavior. The other is purely biochemical, affecting cellular metabolism.
got it. Totally different jobs.
Okay.
Next up, what about those synthetic exercise mimetics? Compounds designed to mimic workout benefits like SLU PP332.
SLU PP332. Yeah, that's a good comparison because both can end up increasing fat burning, but how they do it is different. SLU PP332 works by activating specific receptors inside muscle tissue, these things called ERRs, estrogen-related receptors.
Okay.
Activating those mimics the signaling cascade that normally happens during exercise. It's basically sending a powerful hormonal-like signal that tricks the body into thinking it's exercising, which prompts the muscle to adapt accordingly.
So it's sending a fake exercise signal.
Kind of, yeah. Whereas 5-amino-1-MQ doesn't use that signaling pathway at all. It works purely by improving the cell's underlying chemistry. It conserves NADER, meaning the muscle already has the fuel it needs to respond properly to any signal, whether that signal comes from real exercise or maybe even from a mimetic like SLU PP332. One sends a signal, the other optimizes the fuel supply needed to respond to signals.
Different roles again. Okay, how about MOTSC, that mitochondrial peptide that gets talked about a lot in aging research circles?
V's MOTSC. Right.
MOTSC is fascinating. It acts more like a molecular communication tool. It's a peptide, a small protein, and it often works by changing gene expression patterns. It influences how the cell handles stress, how it regulates its mitochondria.
Yeah.
You can almost think of MOTSC as the coach, giving instructions to the cell on how to manage resources better or adapt more efficiently.
Okay, the coach. So where does 5-amino-1-MQ fit in that analogy? 5-amino-1-MQ is more like the energy drink or maybe the repair crew that fixes the fuel leak. It doesn't give instructions like MOTSC does. It just prevents the molecular waste that would otherwise hinder the cell's ability to follow instructions, whether those instructions come from MOTSC or exercise or other hormones. It enables the response.
Okay. Energy drink, not the coach.
Mm-hmm.
Got it. Finally, maybe the most nuanced comparison, the NADO boosters. Things like NMN or NR, both are aiming to improve NADO levels, right? But they approach it very differently.
This is probably the most critical distinction for a knowledgeable listener to grasp. These NADO boosters, NMN or NR. Boosters like NMN or NR, they are precursors. They work by increasing NAD levels by adding more raw material to the system.
Okay.
They're like delivering extra truckloads of coal to the factory. They build up the stockpile from the input side.
Right.
5-amino-1-MQ doesn't add anything new to the system. It works entirely by preventing loss. It's the maintenance crew patching the hole in the fuel tank, protecting the existing stockpile by blocking that wasteful NNMT enzyme.
Ah, okay. So if the main problem is just that the cell isn't getting enough raw material, maybe a booster is the way to go.
Could be.
But if the problem is that the cell is actively wasting the material it already has, then 5-amino-1-MQ is the specific tool for that specific leak.
Precisely. They target completely different bottlenecks in the same overall energy system. One increases the supply of fuel, the other eliminates the waste of the fuel that's already there.
Which raises an interesting point.
Yeah, the sources definitely suggest the exciting possibility that because their mechanisms are so distinct, they might actually be complementary. You could potentially use a strategy that combines maximizing the input with boosters while simultaneously minimizing the output loss with something like 5-amino-1-MQ.
A kind of two-pronged approach?
Exactly, a more holistic approach to managing NADU levels, and that might be where the future of metabolic intervention really lies.
Okay, we've covered the power, the unique mechanism, how it compares. It's all very exciting science. But as we move towards wrapping up, we absolutely have to ground this excitement in the necessary reality of preclinical research. Starting with that critical question, what about safety, at least in the animal models studied so far?
We must. Yes, and the data we have so far is actually highly encouraging regarding its specificity. And high specificity often correlates with a better safety profile down the line.
Okay.
In the animal studies reviewed, including ones using pretty high doses and regular prolonged administration, the compound was consistently reported as being well tolerated. They noted no visible toxicity.
Right.
And that initial safety finding is, frankly, what allows researchers to even continue pursuing this line of investigation. It's a prerequisite.
That's the foundation, then, for any potential future human research. However, and this is the good however, we have to deliver that essential disclaimer, addressing the preclinical context head on.
Let's be absolutely crystal clear on this point. No human trials have been conducted yet. Zero. All the robust data we've discussed, the fat loss, the cholesterol improvements, the muscle strength gains, the enhanced regeneration, it all comes exclusively from cell culture experiments and animal models, mostly mice. So, therefore, the long-term safety in humans, the appropriate human dosage, and the definitive effectiveness in people, all of that remains completely unknown at this stage.
And it's important to remember why researchers look for that specificity you mentioned earlier, right? Why is that a good sign, even at this early stage?
Specificity is good because it suggests the compound is doing one thing very, very well in this case. Blocking NNMT without acting like a bull in a china shop, without being a systemic disruptor hitting lots of other targets.
Right.
Researchers actively seek compounds that are almost surgical in their action because those typically have fewer off-target side effects. That makes them more promising candidates for eventual clinical development. But that promise requires rigorous, careful, measured investigation through properly controlled human trials before anyone can draw conclusions about using it in humans.
Which brings us neatly to the final, absolutely necessary legal and ethical boundary. This was found explicitly within our source material regarding how these compounds can currently be used or marketed.
Yes, we have to state this clearly. We must adhere to the fundamental regulatory distinction here. Substances at this stage of development are strictly, and I mean strictly, limited to research purposes only. Meaning they are not intended for human consumption, they are not approved drugs, they are not dietary supplements. This distinction is absolutely critical for safety, for regulatory compliance, and frankly, for responsible discussion of the science. We're here to convey the scientific findings, not to endorse or encourage any kind of unauthorized usage. #tag outro.
Okay, so let's wrap up this deep dive for you, the listener, the learner. The really big idea with 5-amino-1-MQ is this. It's a highly selective inhibitor of this enzyme called NNMT. And this isn't about fiddling with appetite or major hormonal signals. It's about correcting a specific biochemical inefficiency inside the cell.
Right. It acts almost like an internal repair mechanism. It revs up the cell's own energy engines by conserving two absolutely critical resources that NNMT would normally just waste. NADO, that key energy currency, and also SAM, the cell's universal methyl donor, that sort of repair and maintenance budget.
And the observed result, at least in these compelling preclinical models, is a clear shift towards metabolic efficiency. We saw significant fat mass reduction without changes in appetite, coupled with these really remarkable improvements in muscle strength and regeneration capacity, all achieved through this very targeted biochemical route. It really forces us to rethink the balance between internal efficiency versus just focusing on external inputs like diet.
Which leads us right into our final thought, something for you to consider. We learned today that blocking NNMT doesn't just save NNAU, the energy molecule. It also saves SAM, that molecule crucial for DNA repair, gene regulation, all sorts of cellular maintenance.
Yeah.
Now, if NNMT levels really do tend to rise with age and obesity, as the data suggests, this implies that maybe the aging process itself involves a kind of systemic, accelerating waste of fundamental resources. Not just energy resources via NADU, but repair and maintenance resources via SAM2, across multiple pathways.
So if NNMT is actively draining both the energy potential and the repair potential as we get older or metabolically stressed, then maybe the most powerful therapeutic lever isn't necessarily just flooding the system with new supplies like more B3 vitamins or NADU precursors.
Maybe not. Maybe it's simply fixing the drain itself. This raises, I think, an important question for all of us interested in this field. How might optimizing waste prevention, the mechanism we see with 5-amino-1-MQ, prove to be a more fundamental, maybe more sustainable long-term strategy for maintaining cellular health, compared to simply trying to increase the supply of precursors like NMN or NR alone? The potential implications of conserving both SAM and NAD simultaneously. It really challenges our current thinking about how we might approach aging interventions. What's more fundamental, boosting supply or stopping waste?
That is a profound thought to mull over, fixing the leak versus just pouring more fuel in. Thank you for joining us on this deep dive into the really cutting edge of cellular science, these fascinating metabolic switches. Stay curious, everyone, and keep asking those deep, fundamental questions.